All right, so this is going to be the second portion of lecture number 8. In the first portion, we talked about the elucidation of the structure of DNA and the mechanism by which DNA is replicated. In this one, we're going to talk about the structure of RNA and the role that RNA plays in two other genetic processes, transcription and translation. So the structure of ribonucleic acid differs ever so slightly from DNA. You can see that in DNA, remember that the D stands for deoxy. So a deoxyribose, a sugar, has no hydroxyl group at carbon number 2, whereas plain old ribose sugar, the type of sugar that's in RNA, has this hydroxyl group at the second carbon. So a very, very subtle difference, but it gives very, very different chemical properties to RNA and DNA. So one of the changes of what I just talked about, ribose versus deoxyribose, so the presence of this hydroxyl group at carbon number 2. The other difference we talked about, or I discussed or presented in the first part of this lecture, in that the nitrogenous base uracil is found only in RNA, whereas the nitrogenous base thymine is found only in DNA. The other thing that I want to discuss in terms of the differences between RNA and DNA is their structure. So the overall model of DNA is that DNA is double-stranded. It's kind of the vast majority of time DNA exists as a double-stranded molecule, and that fact that it's double-stranded makes it extremely stable. So it's yet another reason we talked about the sugar phosphate backbone and that lending itself to the stability of this molecule, and then the second portion that maintains its stability is the fact that it's double-stranded. And then I would say maybe a third reason why it's so stable is that it's complex with many different types of proteins that keep the structure of DNA very stable. The superstructure of DNA very stable. RNA, on the other hand, is almost always single-stranded. There's a couple of kind of rare instances where it can kind of fold on itself and, like, for a split second or two, become double-stranded. But RNA, for the most part, is a single-stranded molecule. In fact, when a cell sees, like, if a cell can sense that double-stranded RNA is inside of it, it actually thinks that it's being invaded by, or infected by a virus, because viruses can have double-stranded RNA. So it's a double-stranded RNA basically for us and even for bacteria is considered a foreign, like, bad things. It's kind of a sign of something's wrong. So RNA, always single-stranded, and it's also very unstable. Interestingly, so RNA doesn't. I should look up what the half-life of RNA is, but it's probably, like, especially in bacteria, as soon as it's translated into protein, it basically gets broken down. It's really a very cheap copy. The way that, the analogy that I like to give, let's imagine that we were a general contractor. We're like this big architecture firm, and we have a contract to build a very, very complex, very expensive skyscraper downtown San Diego. So we have our architects and all of our engineers draw up this very special blueprint that's going to serve as the instruction for all of us to refer to. And we only have one copy. We only have one copy of that very important blueprint. And so we want to keep it safe. We're never going to let it out in the sun or the rain. We're not going to let people borrow it. And we're going to keep it in this very special room, and everyone can come in and refer to it and take notes and look at it. But we don't want it to leave the room because we can't lose it. That's DNA, right? It's our very, very special set of instructions that we need to keep safe and never lose and keep very stable and so on. If we were able to, like, all of our plumbers, our electricians, all of our carpenters, all the people that are building the skyscraper, they can go in and they can make a copy of it. They can put the blueprint on a big photocopier, right? And they can make a copy of it, and then they can take this copy out to refer to when they're actually building the different parts of our skyscraper. Well, that cheap copy that they made and they don't really care about because they can always just get another one, that's RNA. All right, it's a copy of our instructions that we're actually going to use to translate into something that's three-dimensional, or that is kind of, it serves as an end purpose. Of course, our skyscraper analogy, it's our plumbing, our electrical work, and so on. But in our cell, it's our proteins, right? Remember that DNA, ultimately, our genes ultimately code for a protein, and that's what proteins are, the things that do all the work. Remember all the enzymatic functions in metabolism and so on. Why does this it never clicks in the early part? I have to click it like 7 times. Okay, so for our purposes, there's actually many types of RNA. I can think of probably 5 or 6, but for our class, we're going to talk about the 3 main kinds of RNA that are common in all organisms and that are essential for this expression of genes. And that's, of course, messenger RNA, mRNA, rRNA, ribosomal RNA, and tRNA, transfer RNA. And all of these RNA products are made from transcribing a portion of our DNA. So our DNA has a particular kind of sequence, and that sequence serves as a template for transcribing messenger RNA, ribosomal RNA, and transfer RNA. And of course, messenger RNA ultimately gets translated into a protein. So let's talk about these kinds of RNA, and let's talk about ribosomal RNA first. If you recall, all living things have ribosomal RNA, or ribosomes. We also call them, they're little tiny free-floating organelles called ribosomes. We have them, bacteria have them. Any living organism has ribosomes. Now, remember that the structure of our ribosome is very importantly different than the structure of bacterial ribosomes. And this is a difference that we can exploit using different antibiotic medications. So there are antimicrobial medications. I think like tetracycline, for example, I think tetracycline is one of the antibiotics that specifically target the 70S ribosome of bacteria. And this is why we can take tetracycline and it doesn't affect our ribosome, because we have a different structure. Our ribosome is a 40 and 60S subunits that come together to form an 80S subunit. In bacteria, the 2 subunits, we have a 30S subunit and a 50S subunit. And while these are made of RNA, they're also made up, or I shouldn't say they're made up, but they complex with proteins. They complex with lots and lots of polypeptides, proteins, to make this superstructure called a ribosome. And it's only when these 2 pieces of RNA, these 2 pieces of ribosomal RNA get together with proteins and get together with each other that we have this, some people call this a hollow enzyme. So it's kind of 2 enzymes coming together to have some type of enzymatic activity. And this is when we get translation occurring. Remember that ribosomal, I'll talk about this in more detail here in a bit, but ribosomal RNA is a ribosome, and of which a ribosomal RNA is an important part, is where protein synthesis takes place. So where translation takes place. All right, let's talk about messenger RNA, mRNA. Messenger RNA, again, is our copy. And I shouldn't call it a copy because it's not really a copy of DNA. It's a complementary copy of DNA. It's the complement of DNA. It's not an exact replica of DNA. It is it's a code that's based on the instructions and based on the sequence that DNA has. So messenger RNA carries that message from DNA to the ribosome, where protein translation is going to take place. So messenger RNA is the code itself. It's the string of RNA that serves as the basis for protein synthesis, whereas transfer RNA and the ribosomal RNA are involved in the translation mechanism itself. So I kind of like this picture because it's very simple, but it's a schematic of what DNA does, right? DNA serves as a template for its own replication, and it also serves as a template for transcription of messenger RNA, which in turn serves as a template for translation into protein. So so DNA is made up of nucleotides, A, T, C's, and G's, and it codes for a messenger RNA that's also made of nucleotides, A's, A, U, C, and G. And every third code, or every third nucleotide, every 3 nucleotides is a group. And we're going to call that group a codon because that's really our code. Think about any code. We have binary code, right? That's what computers use. We have Morse code. There's all different kinds of codes that we use and we have to have a set of rules on how to read that code. And so the set of rules for reading DNA code or quaternary code with these 4 different nucleotides is called a codon, and they're in sets of 3. And I'll show you how that works in this slide. So here we have our strand of DNA, and you can see the code here, right? So ultimately, how does this code get turned into a protein? That's the question that we're asking. And so this top strand is the one that we're actually going to use as a template for transcription. And we're going to refer to this top strand. I'll talk about this more in a later slide, but we're going to refer to this top strand as the minus strand or the antisense strand. So this top strand is going to serve as a template for its complementary messenger RNA. And you can see that they're complementary because T is complementary to A is complementary to U, C is complementary to G, and so on, right? And in order to read this code, we need to separate them into codons. And so we can basically find our start codon, which is AUG, and every third, right, we're basically kind of grouping them into groups of 3 every few nucleotides after that. So our first codon is AUG. Our second one is AAG. Third one is UUU. And our last one, and the very short, remember proteins are, oh my goodness, thousands of amino acids, maybe tens of thousands. I have to look up what the longest protein or the longest gene or longest protein is, but there are thousands and thousands of amino acids long. Of course, here ours is 4 amino acids long and so on. So, or actually it's only 3 because we have a stop codon. So our messenger RNA is organized into codons. Remember, they're groups of 3 nucleotides, and we have a start codon and a stop codon. And you can see that these codons code for very specific amino acids. In fact, I think that's our next slide. I hope it is. Okay, great. So how do we know this, right? And again, yet another Nobel Prize was awarded for the scientists that figured out ultimately what codons coded for which amino acids. Remember that proteins are made up of amino acids. There's about 20 of them, I think, around 21, 22 of them that serve as the kind of the alphabet for all of our proteins. And you can see them all here: phenylalanine, leucine, isoleucine, methionine, valine, alanine, and so on, right? So all of these amino acids are what ultimately make up not only us, but everyone: bacteria, ducks, dogs, octopi, octopus, the fish, everything, every living thing on this planet not only has the same amino acids, but they also have the same genetic code, which I think is just such a beautiful and mind-blowing idea, that the very thing that serves as a code for all of our proteins and really us is the exact same code that bacteria and these ancient, organisms, the archaea, those original, kind of proto-life forms that were here billions and billions of years ago. Same thing, same code. Pretty cool. So you'll notice that not all of these codons, right, code for an amino acid. So some of them actually code for no amino acid and are therefore what's called a stop codon, right? If you think about it, we have to have a code that tells us where to start and a code that tells us where to finish. And so our start codon is AUG. I always remember that because we start school in August. So whenever we find an AUG on our mRNA, our messenger RNA, that is our start. That's where we're going to actually start protein translation. It also codes for methionine. It's a special kind of methionine that gets inserted at our start codon. And then we're going to go along and we're going to add all the various amino acids that are coded for in our template until we get to a stop codon. And that is our signal that a protein translation is over. So the way that this table works, you can see we start with our first letter. We go down the row until we find our first letter. So let's go back one slide, right? So let's look at AAG, right? Here, this codon codes for AAG. So we're going to go down and find A, and then we're going to go across until we find the second letter, which is A, and then it's going to be one of these, AAG. So we can see that AAG codes for the amino acid lysine. I hope that's right. Great. Yep, lysine. Cool. So that's how our genetic code works, and you can see that there's some overlap here. For example, all four of these codons all code for the same amino acid serine, and we can see that because essentially what happens here is that third codon, that third position, doesn't really matter. Right? It can be a U, a C, an A, or a G, and they all four of those options still code for serine. So let's talk about our very last form of RNA called transfer RNA or tRNA. And this is one of those exceptions I talked about. There were some very rare exceptions where RNA is double-stranded. And you can see that RNA has, so RNA doesn't really look like this in real life. This is kind of a, this is one of my colleagues here at City refers to it as the roadkill. So it's like a flattened form of it. And essentially you can see the sequence of RNA kind of follows this blue line around. And you can see that some of the complementary strands force this RNA to have really a three-dimensional structure. If you think about messenger RNA, we want it to be this nice long code that we can easily read and kind of move down those codons, whereas tRNA has to have this three-dimensional shape to it because it has work to do. It's not just being read, it's picking stuff up and it's attaching, picking up amino acids and transferring those amino acids and putting them on that growing polypeptide chain. So this transfer RNA in real life, in reality, looks like this kind of very weird twisted structure. And I want to point out two very special features of this twisted structure. At the bottom, so sorry why this is doing that. Here you go. At the bottom, at the very bottom of this tRNA, we're going to see a very, very special, very important string of codons, or nucleotides called an anticodon. So the anticodon is complementary to the codon that it's going to read. So in this case, the anticodon here is GGC. So we're going to go back a slide and see what that codon is that it's complementary to. And then the other piece of this tRNA that I want to point out is at the very top, where it attaches to that specific amino acid. In this case, it's attaching to the amino acid proline. So again, let's look at the anticodon, GGC. The codon that this would be complementary to would be CCG, right? So let's go back. Can we go back? There we go. So where is CCG? Try to find it yourself and see if you can agree with me. CCG, right here, and it codes for proline. So in order for this tRNA to recognize this codon, this very special triplet that's found in our message, it has to have that anticodon GGC. And so now this tRNA, for example, this particular tRNA that attaches to the amino acid proline is able to recognize that proline code on our messenger RNA. It's so cool. If you think about just this mechanism is just, it's such an elegant mechanism of building something, literally using a code. I don't know. I geek out on this stuff. Okay. Let me, I'm going to show the video for transcription, and then we'll talk about it. Structural basis, messenger RNA transcription.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA. synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA, called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
All right. Let's go back. So there's again, you can always rewind this video, watch that a couple times to kind of get the hang of it. And let's talk, I want to point out a couple of things that the video either briefly talked about or kind of skipped over. When we're talking about transcription, we want to talk about which strand of the DNA molecule we're actually transcribing. It could be the top or the bottom. And it depends on the direction that we're moving the direction that we're moving, the RNA polymerase is moving, is going to determine which strand it is actually transcribing. So we can refer to the strand that is actually being used as the template as the minus strand. So the minus strand of DNA, in this particular case, we know it has to be the minus strand of DNA, because if we look at our RNA, our RNA is going from a 3' to a 5' end here. And so this RNA, this pink piece of RNA, has to be complementary to the 5' to 3' strand, this bottom strand, the minus strand here in the DNA. It can't be from the top strand, because the top strand is also 3'. As we move from right to left, it's also 3 to 5', and it can't be that way. Remember, any DNA or RNA, any nucleic acid that we synthesize is always going to be complementary and anti-parallel to the nucleic acid that we're building it off of. So there's this alternating sense when we're talking about polynucleotides, so a string of nucleotides together. So we call the plus strand the sense strand, and the minus strand the anti-sense strand. And remember, again, they're always anti-parallel. So it's very easy to figure out which strand of DNA this RNA is being built from, because it's always going to be the opposite direction, right? The anti-parallel direction. So remember that transcription, really the very simple definition of transcription, is the process of making RNA from DNA, specifically messenger RNA. Well, I shouldn't say that. That's not actually true. When we make ribosomal RNA, we make transfer RNA, that's also transcription. But when we're thinking about transcription in terms of the flow, ultimately making a gene, making a protein, or making a protein from a gene, we're talking about making messenger RNA. Remember, we're going to use the template strand, the antisense strand, as the template, and that so therefore because we're using this terminology, we can say that the messenger RNA is always a plus strand. In fact, if we go back to this strand, let's compare the sequence. There we go. Compare the sequence of the pink strand to the red strand. What do you notice? They're the same, right? I mean, the only difference, what's the difference? Of course, that the RNA has U and the DNA has T. So if you ever have to transcribe a portion, like maybe say on the test, if you ever have to transcribe a portion of messenger RNA from a DNA template, don't bother trying to actually, go through each nucleotide. Just rewrite the top, just rewrite the plus strand, right? And then sub out all the T's, sub them out for use. Easy peasy. So in other words, all that to say that the RNA strand, the messenger RNA strand, will always be identical to the DNA, the plus strand of DNA, except for the uracil thymine switch. Remember that, again, we're going to talk about our 3 stages of transcription: initiation, elongation, termination. RNA polymerase is the enzyme responsible. It typically requires a binding factor called a sigma factor to help it find the promoter region. And so initiation begins when the RNA polymerase, the sigma factor, and when the RNA polymerase and the sigma factor come together at the promoter site. It's added in the same fashion as DNA replication in that synthesis happens in a 5' to 3' direction. Nucleotides are always added to that 3' end. And elongation is going to stop. It basically pauses at a termination site. So it's a very specific sequence of DNA that causes the RNA polymerase to kind of pause, and that pause causes it to fall off. And we now have a complementary sequence. We have a complementary piece of RNA based on our DNA sequence. So here's just a nice picture. This basically, I'm not going to spend too much time on this because this is what the video just showed us really quite nicely. I would recommend just rewinding this video back to where that video went over this in moving pieces. Moving parts in, you know what I'm trying to say. Okay, so now let's move on to translation. Again, I'm gonna go off the lecture here and go to the video because the videos just explain it better than I do. I'm modest enough to know this. Let's do, oh no, not that one. Let's do this one is better. Yeah, this one is good. Okay.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
All right. So let's talk about these steps now that we saw the walkthrough. Remember that translation, we're going to take that code that is now in messenger RNA, we're going to take that code and we're going to translate it into a new macromolecule protein. And this happens again in three stages, initiation, elongation, and termination. Initiation occurs when these two subunits, remember our 50S subunit and our 30S subunit, our ribosomal complexes come together to form that hollow enzyme, that 70S ribosomal subunit. So this subunit is going, or this ribosome, I'm sorry, is going to scan along this this transcript until it oh my gosh, can't talk. This ribosome is going to scan along this ribosomal oh, for goodness sake, this ribosome is going to scan along the transcript, the messenger RNA, until it finds its first start codon. Why is this showing elongation? Do I not have a... Oh, I guess I don't. Okay. Sorry. Ah! There we go. Sorry. Okay. So it's going to scan along the messenger RNA transcript until it finds its first start codon. And at that point, we have initiation of translation. Elongation is going to occur, like the video showed, by basically adding amino acids one codon at a time. So we can see here, we can see our first start codon, AUG, is already in the E site. I meant to look up what E stands for. I always thought E stands for exit. If it doesn't formally, you can think of it as exit because this is where the tRNA exits. And then our next start codon, or our next codon on the messenger RNA is CCG, which codes the complementary anticodon is GGC, which codes for proline. And we can see coming into that A site is our next codon UAC, which is complementary to AUG, right? This is our anticodon here in the transfer RNA, and that codes for an amino acid called tyrosine. And so in this way, we have elongation occurring one amino acid, one codon at a time until we get to our stop codon. I think there's 3 or 4 different codons that code for stop. In this case here, we have UAG, and that is telling this stop codon doesn't code for an amino acid and essentially makes the ribosome pause and fall apart and come off of our messenger RNA transcript. But that's okay because we already have our polypeptide chain. And remember, in reality, these things are thousands and thousands of amino acids long. So again, just a quick reminder of our genetic code. To show you, here are the codons that are contained in our messenger RNA. We can see our start codon here, AUG, that also codes for methionine, and then we have our 3 stop codons all up here. I think I already talked about this, right? Translation, I kind of described it using those pictures that were before. Elongation is occurring by adding those amino acids and transferring them over using actually a very interesting and important enzyme called peptidyltransferase. Remember that that amino acids are held together by a special kind of bond called the peptide bond. And so the enzyme that's part of the ribosome that actually makes that peptide bond form is an enzyme called peptidyltransferase. And then elongation continues until we reach that stop codon that arrives at the A site, and that causes translation to stop. This usually involves some type of release factors, which are proteins. So at that stop codon, once that ribosome encounters that stop codon, it pauses, and then these release factors, which are basically proteins that are recruited to the site that help the polypeptide, our protein that's forming dislocate from the ribosome and fall off. Okay, so we're actually going to practice, I think it's going to be in our next, I'm going to break up this lecture yet again, but we'll practice transcription and translation in the next lecture. I'll give you an opportunity to actually take a piece of DNA and transcribe it into RNA and then translate it into protein. But now, very briefly, we're almost done. I want to talk about the differences between prokaryotes and eukaryotes in terms of their molecular biology. What's really cool, I think, about bacteria, remember that bacteria, again, some bacteria double in as little as 15 minutes. So this has to happen at a breakneck speed, and bacteria are competing with one another for resources, for doubling time, for use of nutrients, and so on. And so therefore, the more efficient you are, the greater advantage you have over your neighbors. So in bacteria, the route that they take towards genetic expression is all about efficiency. They want to be very, very efficient and very fast when they're replicating their DNA and when they're transcribing it and translating it. So you can actually get replication occurring, and while that replication is occurring, the transcription machinery can land on a piece of that DNA and start transcription. And while transcription is occurring, ribosomes can land on that transcript and start translating protein. In other words, this can all three of these processes could be happening at the same time in prokaryotes. So that never happens in eukaryotes. Eukaryotes have very, very specific times where each of these process and places, not just times. It's not only kind of separated temporally, but it's separated spatially as well. So you can see that here. So here's our DNA, right? Here's our chromosome of our bacteria. And we see our, what enzyme would this be? This is our RNA polymerase, right? Because it's making our messenger RNA. Here's our code. So this is transcription occurring. And then you can see our ribosomal subunits, our 30 and 50 S ribosomal units coming together to actually start translating this protein based on this messenger RNA. So here we can see transcription and translation happening at the same time. So as soon as this messenger RNA is complete, as soon as this RNA polymerase pulls off at the termination site here on our DNA, these ribosomes are just kind of following along right behind it, so that we now have how many? 1, 2, 3, 4, 5 copies of whatever this protein is, right? That's super, super efficient. That never happens in eukaryotes. Eukaryotes are way, way too complicated to have multiple ribosomes occurring, processing the same piece of messenger RNA as well. So a couple other things that I want to point out. This is a table I think in your book as well. Messenger, again, let's look at our previous. Messenger RNA is not processed in any way. As soon as that comes out of the RNA polymerase, it is now considered, free game for translation into protein. So this is happening pretty much right away as soon as this messenger RNA is transcribed. However, in eukaryotic processes, that messenger RNA has to go through all kinds of processes before it's actually made into a protein. So the first thing, there's several things that happen. The messenger RNA is modified. There's there's modifications made to the 5' end of messenger RNA and the 3' end of messenger RNA to stabilize it. Remember that RNA is a fairly unstable molecule. So what happens in our cells is that that messenger RNA transcript has to get kind of modified to make it more stable so it lasts a little bit longer. Also, don't forget that eukaryotic genes, our genes, contain lots of non-coding regions called introns. All of those introns have to get cut out and removed from our messenger RNA transcript, and that happens before it's taken out of the nucleus. So the next thing that happens to the messenger RNA in a eukaryotic cell is that it has to get exported out of the nucleus. It has to go, remember, our ribosomes are in our cytoplasm. They're outside the nucleus. So our messenger RNA has to get processed, to introns taken out, and then it's exported into the cytoplasm where ribosomes are waiting to finally translate it. Another kind of cool thing about bacterial genes is that they tend to be polycistronic, which means a cistron was an old word for a gene. And so one messenger RNA transcript can actually code for lots of genes. And we'll see that when we talk about gene regulation here in a couple lectures. But that never really happens in eukaryotic genes. Eukaryotic genes, the messenger RNA is usually monocistronic. In other words, that messenger RNA, one mRNA, will code for one protein. Whereas in bacteria, one messenger RNA could code for 5 different proteins. In other words, there's multiple start codons along the way of that messenger RNA. And as soon as that ribosome finds a start codon, it's going to just start translating. So let's kind of think about this in terms of looking at a transcript, or our DNA and our transcript and our messenger RNA. If a prokaryotic gene is, let's say, 300 base pairs long, and let's ignore the fact that we have a promoter region and so on, the messenger RNA transcript is also going to be 300 base pairs long. However, we can't really predict how many amino acids are in the protein because it could code for multiple proteins. It could code for one protein, or it could code for 4, 5, 6 proteins. So we don't necessarily know how long or how many amino acids would be in any given protein. But now let's contrast that to a eukaryote, right? In eukaryotes, we have our DNA, and we can see that there's exons and introns encoded in that DNA. So when this DNA is transcribed into a messenger RNA, we can modify it by putting a cap on the 5' end, and then on the 3' end, we put a poly A tail on the I believe it's on the 3' end. And then we have to take out these introns, right? The introns get literally just like cut out by very special enzymes that can cut DNA and stitch it back together. And now when this messenger RNA gets exported out into the cytoplasm for protein translation, we have no idea really how long this transcript is. But we do know that if the gene is 300 base pairs long, that the protein would be roughly 100 amino acids long, because we know that this codes for one protein. So some important differences between prokaryotes and eukaryotes. All right, so a nice little review process that might help you in kind of remembering the highlights of this lecture would be to make yourself a table that talks about these 3 processes. And remember that each of these processes had 3 stages: initiation, elongation, and termination. And you can review the major steps of each. And what I would recommend you do in elongation is jot down the important enzymes that are responsible for elongation, and in the termination column, put where termination actually ends. All right, I think that does it for this lecture. Yep, I'm going to go ahead and pause it there. Again, another 40-minute lecture. These are longer than I would have thought. And in this next lecture, I'm going to talk about mutations and the consequences of mutations.